Hi, and welcome to this series exploring landmark experiments in protein science based on my book of the same name, which can be purchased on Amazon, and that link is in the description. In the last video, I talked about the discovery of cells and found that little was known about the cell and its contents even up to the 1900s. Cells were found to be composed of a cell membrane, a protoplasm, and a central nucleus, which contained stick-like structures known as chromosomes, but no one really knew the function or the importance of this substance, although it looked like it had something to do with the inheritance of traits. And so in this video, we'll turn our attention to the discovery of proteins and enzymes. The discovery of protein dates as far back as 1747 when Beccarii used a variety of solvents to isolate a substance from wheat, which resembled something that did not seem possible to extract except from animal matter. Since the substance was similar to egg albumin, which is basically egg white, he referred to it as a plant albumin, although it was later renamed wheat gluten from the Latin for glue due to its sticky nature. Parmentier repeated these experiments in 1773 and published the precise protocol. He added water to 2 pounds of flour to make a paste, over which he trickled a steady stream of water as long as the runoff was of a milky color. This left behind a few ounces of a substance which he described as being similar to a tenacious membrane, elastic, insoluble in water, sticky and adhered well to dry material, without flavor, having an odor of glue and a yellowish color. He and others characterized the substance and determined that not only were they found in a number of different plants and flowers, but they were also in animals, and they all consisted of the same chemical elements C40, H62, N10, O12, though small amounts of sulfur or phosphorus were also found in some samples. As more and more similar substances were discovered, they were classified in terms of their solubility in various solutions. So albumins were proteins that were soluble in water, coagulated in heat, and remained dissolved when added to a half-concentrated solution of ammonium sulfate. In contrast, the globulins were classified as proteins which were insoluble in water but soluble in neutral salt solutions. These were further divided into the vitellins, which were soluble in saturated sodium chloride, and myosins, which were insoluble in this solution. The prolamines were the alcohol-soluble proteins, and finally, the glutenins, of which gluten was the only known member, was a category reserved for proteins which could not be dissolved in neutral or saline solutions or in alcohol. In 1839, the term protein was suggested by Jacob Berzelius to describe these new substances. Berzelius writes, "The word protein that I propose to you for the organics oxide of fibrin and albumin, I would wish to derive from proteos, which is Greek for standing in front or in the lead, because it appears to be the primitive or principal substance of animal nutrition that plants prepare for the herbivores and which the latter furnish for carnivores." In one of his textbooks from the mid-1800s, Mulder described the importance of proteins as follows. In plants as well as in animals, there is present a substance which is produced in the former, constitutes the part of the food of the latter, and plays an important role in both. It is one of the very complex compounds which very easily alter the composition under various circumstances, and serves especially in the animal organism for the maintenance of chemical metabolism, which cannot be imagined without it. It is without doubt the most important of all known substances of the organic kingdom, and without it, life on the planet would probably not exist. It is found in all parts of plants, as well as in the animal body. Animals draw their most important proximate principles from the plant kingdom. The herbivorous animals are, from this point of view, no different from the carnivores. Both are nourished by the same organic substances, proteins, which plays a major role in their economy. Initially, proteins were thought to be very simple molecules, since all known molecules at the time, like water, ammonia, sodium hydroxide, and sodium chloride, were simple molecules, and there was no reason to think that proteins would be any different. But in the mid-1800s, Thomas Graham found that proteins were indeed different. In his experiment, he pipetted a salt solution at the bottom of a large column of water, doing so slowly and carefully as to not disturb the water. After a certain amount of time, usually in days, he removed the water one layer at a time from the top down, removing a total of 16 layers, and assessed how much salt there was in each layer by letting the water evaporate. As a result, he was able to determine how far each type of salt could diffuse. The more salt present in the layer farthest away from the bottom of the jar, the more diffusible the salt. Using this technique, he found that, in contrast to salt solutions, most proteins diffused very little in water, remaining mostly at the bottom of the jar or in the lower strata. Next, Graham investigated the degree to which solutes and solutions diffused across a membrane by placing a piece of wetted parchment paper at the bottom of a small glass bell jar and added a solution on top. He placed the jar over a large volume of water and the contents were allowed to diffuse into the water over a period of time. He called this novel type of diffusion dialysis, from the Greek dia, through, and lysis, loosening or splitting. His results showed that although various salts could easily diffuse through the membrane and into the water, proteins such as albumin and gelatin were significantly inhibited. For example, albumin diffused 1,000 times less than table salt, which was his standard for comparison since it completely diffused through the membrane. In 1752, the French scientist René-Antoine Ferchaud de Réaumur wanted to know if some birds could digest meat without a gizzard, which is an organ found in many birds that grinds food using pebbles. To study this, he attached a piece of meat to the end of a small tube which he inserted into a bird's stomach through a fistula in its belly, and because it was suspended, the grinding action of the stomach on the meat was prevented. Surprisingly, though, he found that the meat was completely digested within 24 hours, which indicated that a chemical process was at play. Later in the 1700s, the Italian Spallanzani showed that this also applied to other species, including humans, and he showed this by using his own gastric juices. In 1833, Payen and Perzos found that macerating germinating barley, oat, and corn in cold water yielded a clear liquid which contained an active principle which could convert a starch into sugar plus a small amount of nitrogenous matter, colored substance, and a quantity of sugar proportionate to the degree of germination. They found that adding alcohol to this liquid effectively precipitated the active ingredient, forming an insoluble flocculent deposit which could be filtered and dried at low temperature. They named the substance diastase from the Greek separation, since it could effectively separate the inner starch from seed coats. Importantly, they determined that keeping temperatures well below boiling, and they stressed even below 75 degrees Celsius, was crucial to avoid losing activity. To test their extracted diastase, all they had to do was add it to flour grains, such that 1 part by weight is enough to render soluble in hot water the internal substance of 2,000 parts of dry starch in about 10 minutes. In the following years, a number of other substances were found to change one substance into another. Emulsin, purified from almonds, could convert amygdalin to benzaldehyde, and pepsin, which was isolated from stomachs, could curdle milk. And so ferments was used to describe these substances, since they behaved somewhat like alcoholic fermentation, which converts sugar into alcohol. In 1836, Berzelius wrote that this new force, which was unknown until now, is common to organic and inorganic nature. I do not believe that it is a force entirely independent of the electrochemical affinities of matter. I believe, on the contrary, that it is only a new manifestation, but since we cannot see the connection and mutual dependence, it will be easier to designate it by a separate name. I will call this force catalytic force. In 1876, the term enzyme, meaning in yeast, was proposed by Wilhelm Kuhn to describe proteins that accelerate chemical reactions without being consumed in this reaction. In 1884, Emil Fischer subjected his large library of sugars, both synthetic and natural, to a variety of yeast strains to determine if they could be fermented, that is, if they could be turned into alcohol with the release of CO2 as a byproduct. But some of these sugars were in very limited quantities, so he devised a small fermentation tube in which sugar was added to yeast, and barium hydroxide, which becomes turbid when exposed to CO2, was added into a trap. So if fermentation occurred, he could see the barium hydroxide become turbid. His experiments showed that, in all cases, even when the sugar is not fermented, a small amount of carbon dioxide evolves, which covers the surface of the barium hydroxide with a thin layer of carbonate. Since this phenomenon occurs even when no sugar has been added to the solution, it is obviously caused by the small amount of carbohydrate present in the yeast itself or the extract. The situation is quite different when the material is readily fermentable. The barium hydroxide is not only becoming strongly turbid, but is neutralized. Intermediate cases are these where material has to be brought into a fermentable state first. Fermentation proceeds slowly, yet here too the amount of carbon dioxide developed is always large enough that one cannot be in doubt about the real occurrence of fermentation. He also found that a given strain of yeast could only ferment certain types of sugar, even when two sugars were isomers. And so isomers are molecules with the same elements present in the same amount, but where the elements are arranged differently. For example, these are some of the isomers of C6H12O6. Notice how D-galactose and D-talose only differ in the orientation of one hydroxide group, and how glucose and fructose differ by the position of the carboxyl group. And since this was the case for a number of enzymes that he studied, Fischer used the term lock and key to describe the relationship between enzymes and their substrate, an analogy still in use today. The restricted action of the enzymes on glucosides may therefore be explained by the assumption that only in the case of similar chemical geometrical structure can the molecules so closely approach each other as to initiate a chemical action. To use a picture, I would like to say that enzyme and glucoside have to fit together like lock and key in order to exert a chemical effect on each other. The finding that the activity of enzymes is limited by molecular geometry to so marked a degree should be of some use in physiological research. By the 1890s, the requirements for fermentation were fervently debated and involved two sides. The enzyme theory of fermentation, as the name suggests, proposed that enzymes were responsible for fermentation, like when diastase converts starches into sugars, which occurs in the absence of cells. In contrast, the vitalistic view proposed that intact living organisms, such as yeast, were required for fermentation to occur, since fermentation was known to stop when yeast cells were destroyed in a mixture. A minor few, such as Liebig, held a middle ground. It may be that the physiological process stands in no other relation to the fermentation process than the following. A substance is produced in the living cells which leads to the decomposition of sugars and other organic molecules. The physiological process would in this case be necessary to produce this substance, but would stand in no further relation to the fermentation. This debate was finally put to an end by two German brothers in 1897, Hans and Edward Buchner, when they investigated if there was any special action attributable to the contents of the yeast cell during fermentation, expecting that there wouldn't be since they were partial to the vitalistic view. First, they needed yeast cell extracts, which at the time was obtained using harsh chemicals which could damage the extracts. So they outlined specific conditions under which extraction had to occur, but resolving these issues did not come easily. My first attempts to burst the cell membrane of yeast by freezing to minus 16 degrees Celsius and rapid thawing did not succeed. The membrane is much too elastic for it to be broken in this way. Direct grinding in a mortar had as little success because the pestle could not get a purchase on such elastic stuff. Eventually, they settled on a lesser-known method of crushing cells with sand, and Hans's assistant, Martin Hahn, devised an effective method to extract proteins from cells. 1,000 grams of yeast were ground for a few minutes with 1,000 grams of quartz sand and 250 grams of diatomaceous earth, a natural soft rock composed of fossilized diatom, until it became moist and plastic and to which water was added. The mixture was then placed in a cloth and compressed under 400 to 500 atmospheres of pressure, which could yield a total of about 500 ml of juices, could then be filtered to yield a clear but opalescent yellow liquid with a pleasant yeasty smell. Unfortunately, their extracts lost their enzymatic capabilities quickly, so they tried to stabilize the solution by adding a variety of different preservatives like antiseptics, concentrated salt solutions, glycerin, and a sugar solution. When Edward visited the lab one day, he noticed bubbles coming off of one of the sugar solutions and assumed it was due to fermentation by a small amount of yeast left over in the solution. But as they looked into this, they found that this was not the case. First, it had already been shown that these extracts contained too little yeast to account for the fermentation observed. Second, there was no decrease in the extract's fermenting capabilities even after yeast cells were filtered out. Third, the addition of antiseptics to kill any yeast cells that might be present in the extracts didn't decrease its activity. And finally, yeast whose growth had been inhibited could still ferment sugars. And so these experiments confirmed that there is no yeast in their fermenting solutions, and so they concluded that it is much more likely that the agent of the juice which is active in fermentation is a soluble substance, doubtless of an albuminoid substance. This may be designated as zymase. And this finally put to rest the debates between the vitalistic view and the enzyme theory of fermentation. Even though it was generally agreed that no one had won nor lost, fermentation was a chemical process but using molecules extracted from living organisms. Looking back on this period, Buckner stated that the differences between the vitalistic view and the enzyme theory have been reconciled. Neither the physiologists nor the chemists can be considered the victors. Nobody is ultimately the loser, for the views expressed in both directions of research have fully justified elements. The difference between enzymes and microorganisms is clearly revealed when the latter are represented as the producers of the former, which we must conceive as complicated but inanimate chemical substances. And so to summarize, by the early 1900s, proteins were found to be complicated molecules with unknown functions. However, one type of protein, enzymes, are synthesized by living cells and can catalyze the conversion of one type of molecule into another, such as occurs when sugars are fermented to alcohol, and where the interaction between the enzyme and its substrate is as specific as a lock and key. And so this brings this video to an end. I hope you enjoyed it. And if you did enjoy it and would like to know more about this topic, please check out my book, Landmark Experiments in Protein Science, using the link in the description. If you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video on the discovery of protein subunits and the elucidation of the structure of proteins.